So thank you all for coming tonight. I'm Tom Zinnan. I work here at the Biotech Center. I also work for the Division of Extension in Wisconsin 4-H. On behalf of those folks and Wisconsin Alumni Association and the Science Alliance of UW-Madison, thanks for coming to Wednesday Night Lab. We do this every Wednesday night, 50 times a year. Sometimes I throw the little in and sometimes I don't. Tonight, it's my pleasure to introduce to you Carol Van Hulle. She's with the Alzheimer's Disease Research Center. She was born in Minneapolis, Minnesota, and she went to high school in the suburb there called Brooklyn Park, and then went to Augsburg College, which is also in Minneapolis. Then she went to the University of Colorado to pick up her PhD in psychology. She came here to UW-Madison to do a postdoc in the Department of Psychology. I believe you have stayed since, is that correct? And about 3 years ago, she started working with the Alzheimer's Disease Research Center. Tonight, she's got an intriguing question. What do we know and how early can we know it? Exploring biomarkers related to Alzheimer's disease and other research that's being done here at UW-Madison. So would you please join me in welcoming Carol Van Hulle to Wednesday Night at the Lab.
Is this working, or do I need to change it? Are we good? We're good? All right, thank you. So my name is Carol Van Hulle, and I am really excited to be here. When Tom asked me, I think I actually jumped up and down. I was that excited. Probably not the most professional way to respond. But I remember running across a lecture from Wednesday Night at the Lab when I first came here as a postdoc. I was watching PBS while eating my breakfast cereal, and here was this amazing program. And I thought, oh my God, it would be so much fun to be a part of that, and now I am, which is great. So I am currently a staff scientist and a statistician working with Dr. Cindy Carlson, who's a geriatrician in the VA, and she's also director of the Wisconsin Alzheimer's Institute and PI, or co-PI of the Alzheimer's Disease Research Center, among her many hats. As Tom said, I actually got my PhD in psychology with an emphasis in quantitative genetics, and I came here to study child development for many years. And then a few years ago, I had, a mid-career change crisis, whatever you want to call it, and I moved over to the way across from the Wasteman Center to the hospital to work with the Alzheimer's Disease Research Center and study the other end of the lifespan. And tonight I want to share with you some of the research that we're doing at UW to improve early identification of Alzheimer's disease. Tom wrote a really touching anecdote about his experience with dementia. I don't know if everybody got to see that, but this is a very feared disease. It is a disease that has a tremendous personal and public cost, and I'll talk a little bit more about that later. And so I really wanted to share with you some of the things that we're doing here to get us closer to the day when we can eventually identify somebody at a point where we can intervene and either slow the progression of Alzheimer's disease or even stop it altogether. So this is kind of my overview slide. I want to start by talking a bit about what is aging, what is dementia, and what is dementia due to Alzheimer's disease. I want to describe the two main projects that I'm involved with, which are the Wisconsin Research for Alzheimer's Prevention and the Alzheimer's Disease Research Center. And I'll highlight some of the, or how we're using biomarkers to better understand preclinical Alzheimer's disease, and I'll describe what I mean by preclinical AD a little bit later on. And I also want to highlight some of the other research that's being done through our main RAP and ADRC cohorts. I want to end by briefly touching about on what you can do for yourself and to further AD research. Each of these topics could be a series of lectures all on their own. So I'm giving you really just a very broad overview. I want you to imagine yourself flying over the landscape of Alzheimer's disease research and getting just a kind of this 10,000 foot view snapshot. I think our group is small enough here that if I've said something and you don't quite understand what I said or you're confused about something, please go ahead and raise your hand and I'll try to answer your question. But if you are wondering about something that I haven't touched on yet, maybe try to hang on to that until the end. I might address it as we go along, and if I don't, you can always ask me about it at the end of the presentation. Sound good? All right. This is my obligatory facts and figures slide. In Wisconsin, there's about 110,000 people who are diagnosed with dementia due to Alzheimer's disease, and nationally that figure is 5.8 million Americans. And this is a disease, as I said, that affects whole families and really whole communities. In Wisconsin, 193,000 people are providing care for someone with dementia. Nationally, caregivers of people with Alzheimer's or other dementias provide an estimated 18.5 billion hours of unpaid assistance. So again, this has tremendous private and public costs. And there's an entire field devoted to research on caregivers themselves. It's very difficult to be a caregiver of someone with dementia, and if you happen to be a caregiver or know somebody who's a caregiver of someone with dementia, it's really important to reach out and get help, and I'll talk about some resources later on. As the population ages, we expect to see many more cases of Alzheimer's disease, and it is currently the 6th leading cause of death. So what is normal aging, what is dementia, and what is Alzheimer's disease? I decided to take this middle question first. What is dementia? This is simply a general term for memory loss and other types of cognitive impairment that interfere with daily life. Some of you might remember terms like pre-senile dementia or senile dementia. Those are the same thing. It just depends on whether you experienced impairment before age 65 or after age 65. And there are a lot of roads to dementia. It looks like I don't quite have the most updated version of my slides. I don't know how that happened, but that's okay. This pie graph here is kind of giving you an idea of the prevalence of these different types of dementias. There's Parkinson's, dementia due to Parkinson's disease. Frontotemporal lobe dementia is one that starts early, so in about the mid-40s, and it's characterized by neurodegeneration in the frontal lobe, which can sometimes cause impulsive behavior. So this is one that sometimes comes up in the news where somebody starts like a crazy gambling habit. There's Lewy body dementia, and Lewy bodies are a protein that can form in the brain and cause dysregulation. Vascular dementia is due to brain damage caused by strokes or other processes that impair blood flow to the brain. But by far the most common cause of dementia is Alzheimer's disease. It's the big fish in the pond. Either alone or mixed with other types of Alzheimer's disease, it's responsible for around 70% or 2/3 of all dementias. So I mentioned AD is the most common form of dementia. Yeah. It's neurodegenerative, which means we see a loss in structure and function in the brain, which causes impairment in thinking and memory skills. It's progressive. So it's, there's a progressive loss of ability, and this is sometimes referred to as insidious onset. It's irreversible. We do not have any known cures for Alzheimer's disease, and the treatments that we do have don't get at the causes of Alzheimer's disease. They really try to ameliorate some of the symptoms, and they don't even do that particularly well. Biologically, Alzheimer's disease is characterized by plaques and tangles that form in the brain, as well as the loss of neurons, and I'll go into greater detail about that in a moment. So what is normal aging? Well, as we age, we all experience some declines in our thinking capabilities. It may take longer to learn and recall new information. We might find that we have trouble finding words or remembering names, and these are actually two things that people get very worried about. These are very salient symptoms. And a lot of times people will come into a memory clinic because they're starting to have difficulty with names and words. But I remember one of our geriatricians, Nite, talking about this and saying that if you forget a word or a name, but it comes back to you, maybe hours, maybe even days later, that's perfectly normal. When you want to be concerned is when that word or that name is just gone and you can't get it back. But the bottom line is normal aging means that we're still able to maintain our daily activities, engage in social activities and our hobbies, and pay our bills and do all the things that we're supposed to do as adults, maybe with some modifications. This slide is taken from the Alzheimer's Association, or Alzheimer's Organization website. It's alz.org, and they have a ton of great information. But basically, when cognitive changes become a concern is when new problems arise with memory and thinking, and when these problems start to interfere with daily life. So when we see difficulty completing routine tasks, when a person is confused about the time or the place, or when they have trouble, word-finding trouble, or have decreased judgment, we might get concerned. And often with these changes in cognitive ability, there's changes in mood and personality as well. So a person might become depressed, or they might become apathetic and just not care that these changes are taking place.
Excuse me.
Yeah.
How are these different than regular or other kinds of dementia?
They're not. Like I said, there's many forms of dementia. Some other types of dementia, like the Lewy body dementia, has different types of behavioral characteristics that come with it, and different changes in personality. But this is pretty generic.
So that's pretty much for dementia, not just Alzheimer's.
Yeah, right. Thank you. So I mentioned that dementia is not a part of normal aging. However, aging is the biggest single risk factor for developing Alzheimer's disease. Onset before age 65 is pretty rare, and we call this early onset Alzheimer's disease. The more common late onset Alzheimer's disease, sorry, let me back up. The incidence of late onset Alzheimer's disease doubles every 5 years after the age of 65. So by the time we get to people who are 85 and plus, some estimates claim that over almost half, 45% of people over the age of 85 are estimated to have Alzheimer's disease. And that group, by the way, 85 plus, is actually our fastest growing segment of the population, which is one reason why we expect to see so many more cases of Alzheimer's disease going forward in the future. Other risk factors include genetic risk factors. So many of you, I'm sure, have heard of the allo-lipoprotein E gene. This has a variant called epsilon 4. This doesn't cause Alzheimer's disease, but it does pretty consistently increase the risk for Alzheimer's disease. Gender is a risk factor. Women are more likely to develop Alzheimer's disease than men, and the reasons for that are not entirely clear. Part of it is due to the fact that women live a little bit longer than men, but that doesn't appear to be the entire story. Race is a risk factor. African Americans, Native Americans, and Hispanics are diagnosed at a higher rate at all ages than people of European ancestry. Excuse me. And no one knows exactly why. So here in Wisconsin, Dr. Carrie Gleason and others are working very hard to increase the number of study participants from these traditionally underrepresented groups, so that we can close both the knowledge gap that we have on what biomarkers look like in people from underrepresented groups, but we can also start to diminish the health disparity between these groups and people of European descent. So for clinical purposes, the focus is on behavioral changes seen in the person. If a person or their family notices some of those symptoms that I talked about, a physician will probably do some cognitive testing and a neuropsychiatric evaluation, and they might look at a brain scan or do a genetic test, but they might not. These things are quite expensive and often not covered by insurance, and people don't always have the ability to pay for them themselves. For research purposes, the National Institute of Aging and the Alzheimer's Association have proposed a framework for studying Alzheimer's disease that focuses on the underlying brain changes associated with Alzheimer's disease. So here I'm showing a piece of brain tissue that's been stained to show plaques in that kind of brown blob, and then neurofibrillary tangles are shown in the blue squiggles. And that's a big part of the research that I do here at the University of Wisconsin and that my colleagues do. So let's look at this another way. In this picture, the black line shows how we expect thinking abilities to change with normal aging. It goes down a little bit, as I mentioned. But a hypothetical person on this red line, sorry, this is kind of washed out, is in the process of developing Alzheimer's disease. So you can see there's a progressive decline in cognitive abilities. At some point, this person may experience mild cognitive impairment, and that's defined as impairment on a standardized test in one or more cognitive domains, usually memory. But they're able, for the most part, to engage in activities of daily living. Is anybody watching This Is Us? a handful of you. So one of the main characters in that show has mild cognitive impairment. That's one of their main storylines. So I'm like super tuned into it right now. As eventually this person will progress and continue to decline in their abilities to the point where they get a diagnosis of Alzheimer's disease. Right? So that's what we see outwardly. Inwardly, this decline in cognitive abilities is accompanied by accumulating brain changes that are characteristic of Alzheimer's disease. And there's this period here between these two bars where a person's accumulating these characteristic brain changes, but they're not actually showing any diagnosable symptoms yet. And that is kind of our sweet spot. That's what we call preclinical Alzheimer's disease. So I want to go a little bit more into what these characteristics are. So as I mentioned, there are 3 consistent features of Alzheimer's disease. And this is often how Alzheimer's disease is distinguished from other types of dementia, going back to that person's question. Alzheimer's disease is accompanied by these amyloid plaques, or is defined by amyloid plaques, tau neurofibrillary tangles, and eventually neurodegeneration. Neurodegeneration is in parentheses because that's not specific to Alzheimer's disease. It's just kind of an outcome of these other issues. And we see neurodegeneration in a lot of different disorders. So I want to focus on amyloid first. So if we took this little slice and blew it up into this nice illustrator's image, these brown blobs represent the amyloid plaques. Amyloid refers to a protein called beta amyloid. And this is a protein that we make naturally in our brains. Its function isn't completely understood, but we know that we make it in response to injury to the brain. There are many, many forms of amyloid. It is a super complicated protein, but there's a particular variation called beta amyloid 42, or just A beta 42. And this is a product of an abnormal cleavage of the amyloid precursor protein. This results in a misfolded protein that becomes very sticky. Usually when we have a protein that's not functioning properly or we don't need it anymore, it gets cleared out of the brain through the cerebral spinal fluid or through the vascular system. But A beta 42 is so sticky that it doesn't get cleaned out from the brain. Instead, the proteins start to accumulate and form into dense mats and then eventually into these plaques outside of the neurons. And these plaques can be very toxic to the surrounding neurons, and they can trigger other abnormal processes in the brain. And then the neurofibrillary tangles are shown as blue squiggles. These form inside the neurons. Excuse me. Tau is a protein that stabilizes microtubules in the neuron. And it has a role in aiding communication between neurons. So normally these proteins are nice and straight and orderly. And when they become phosphorylated or misfolded, instead of a nice straight microfilament, you get these wavy tangles that get all tangled up with each other. And then the nerve cells can no longer communicate as effectively. And tau in the presence of amyloid really appears to be kind of the sufficient conditions for producing Alzheimer's disease. And then finally, we see eventually a loss of neurons and overall brain atrophy in people who have Alzheimer's disease. On the left is a brain from a healthy person, and on the right is a brain from someone with Alzheimer's disease. And you can see that on this right-hand side, the ventricles in the middle are enlarged, and the overall size of the brain is shrunken down. And if we took a little slice, we might actually see fewer neurons than we would see if we took the same slice in a healthy, in the brain of a healthy person. We might see some damage to those parts of the neurons that communicate with each other. So historically, and I'm sorry I don't have my most recent slides, because I had a picture of Dr. Alois Alzheimer and his patient, August Dieter. And I think I have it later on. I can show that to you. But historically, in order to study these brain changes, a researcher would have to wait until somebody passed away and then look at slices of their brain. And we still do that. we have a brain donation program. It's a really important part of our research, and we are very grateful to all the people who are participating in our brain donation program. And actually, a definitive diagnosis of Alzheimer's disease is not made until there's a postmortem pathology report. But with advances in technology, we don't have to wait until somebody has died in order to at least take a peek inside their brain and start detecting some of these changes that accompany Alzheimer's disease. So the two main methods that we use to look for accumulation of amyloid plaques and tau tangles is through positron emission tomography and by measuring proteins in cerebral spinal fluid. PET imaging can point to physical changes in the brain, and measuring proteins in the cerebral spinal fluid helps us understand some chemical changes that are taking place in the brain. By using these methods, we have evidence that there is a very specific temporal order to these brain changes. So we see amyloid plaques developing before we see tau tangles start to accumulate in the brain, and all of that happens before we see signs of neurodegeneration. And all of this can happen sometimes decades before anyone shows diagnosable symptoms of mild cognitive impairment or Alzheimer's disease. So I want to pivot now to research that we're doing at the university, because one of our overarching goals is aimed at identifying Alzheimer's disease as early as possible in this preclinical phase before symptoms arise, diagnosable symptoms arise. Our other main goal is to identify factors, modifiable risk factors that are modifiable factors that could potentially reduce the risk of Alzheimer's disease. So the Wisconsin Registry for Alzheimer's Prevention started enrolling participants back in 2001. It's been going on for a long time. We have a total of about 1,500 participants, although not all of them are active right now. The Alzheimer's Disease Research Center started in 2009, and we are one of 30 ADRCs throughout the United States, and we have about 1,000 people in that study. Both of these studies are still enrolling participants. RAP enrolls people who are cognitively unimpaired between the ages of 45 and 60. And the Alzheimer's Disease Research Center enrolls older adults who are unimpaired, as well as people who already have a diagnosis of mild cognitive impairment or Alzheimer's disease, so we can compare what these biomarkers look like at different phases of the disease. Both studies are longitudinal. Impaired participants are asked to come in every year. People who do not have impairments come in every other year. And we do 2 visits. We have a biomarker visit where we get some imaging measures and hopefully a lumbar puncture. And in the other visit, we do extensive cognitive testing. We ask a lot about people's lifestyle, their health history, their medication use. We get blood draws. We get a crap ton of information. Both studies are volunteer-based. So we get our participants through referrals, often from healthcare providers, but we also get referrals from friends and family, other people who are already in the study. We've had some people tell their neighbor, come join my study with me. And we get volunteers through public outreach with some of our partner organizations. There we go. So very briefly and simplistically, the way PET scans work is that we inject a person with a radioactive tracer that binds to the protein that we're interested in. And then we wait a half hour to an hour for that tracer to be taken up into the brain. And this allows us, and then we measure how much of that tracer has bound to the protein that we're interested in. If it doesn't bind to anything, it just gets cleared out and we don't get any signal. So by looking at the amount of signal, we can quantify, for instance, how much amyloid is in the brain. So here's an example of 3 tracers. I believe this is all on a single individual, but I'm not positive about that. On top, this is a tracer called Pittsburgh compound B, and this binds to amyloid. So in this particular image, yellow are areas where there's a lot of amyloid plaques, and red and gray are areas with less amyloid. The middle figure is a tracer for tau tangles. And again, the yellow areas are areas where there's a lot of tau tangles, and in gray there's fewer tau tangles, and you can see that there's more amyloid in this brain than there is tau. Again, going back to that kind of temporal ordering. And then we often measure neurodegeneration by looking at glucose uptake in the brain. And the idea here is that if the neurons aren't working properly, or if there's fewer neurons, the brain doesn't need as much energy, and we'll actually see less glucose uptake, as shown, I think, in dark blue here. So the advantage of PET scans is that they reveal physical changes in the brain, and they show us where those changes are taking place. And this is really important, especially for tau. Tau develops in a very predictable pattern. So we start to see tau tangles in the entorhinal cortex, which is kind of in the temporal lobes, before it begins to propagate into other areas of the brain. But it can be expensive. And we can only measure a few proteins at a time because each protein that we're interested in requires a separate tracer. But one way we use this information is by grouping people according to the changes we see in their brain. So during normal aging, we don't see any significant accumulation of amyloid or tau or neurodegeneration, and this is labeled A-, T-, N-. Oh, goodness. But during this preclinical phase, when people are asymptomatic, so they don't have signs that would lead to a diagnosis of MCI or AD, we can already detect significant levels of amyloid and tau, and sometimes we can even start to detect neurodegeneration before people are showing diagnosable symptoms. And this kind of way of grouping people is part of the framework that was developed by the National Institute of Aging and the Alzheimer's Association for using biomarkers to stage disease before we see those outward symptoms. So here's an example of how we use this data from my colleague, Toby Betthauser. In this paper, which was recently published, people were divided into groups based on PET imaging measures of amyloid plaques and tau tangles. So the lower left-hand quadrant shows people who do not have any signs of amyloid or tau. And in the, that's supposed to be gray. And then the upper right-hand quadrant there where it says A+, T+, that's supposed to be orange. Those are people who have both amyloid and tau. On the y-axis, we have a measure of cognition. This is a composite measure, so it combines executive function, working memory, and something else. I can't remember it right now. And we measure cognitive ability across several different study visits. So each line here is a single person. So you can see that some people have like 2 or 3 measurements, some people have 4 measurements. And you can see that there's a lot of bouncing around, but overall, the people in this A+, T+ group are showing much steeper declines in cognition than people who aren't showing signs of amyloid or plaque. And that's kind of shown over here on the right as well, where we've imposed these nice pretty lines on top of this scatter plot. Each of these dots is an individual. We can also look at how quickly people are developing plaques in the brain. This is an extremely recent paper published by my colleagues Rebecca Kosek and Toby Betthauser again. And this is based on longitudinal PET images. So before we were looking at multiple cognitive measures, now we're looking at multiple PET images over time. And these are PET images of amyloid. And people here are grouped according to how much change we see from one PET image to the next. So people in blue are what we call our fast accumulators. So that is, we see a lot of change in amyloid, a lot more amyloid on subsequent PET imaging than we see on the first image. People in green and red are slower accumulators. And then that big line of gray, those are people who don't have any signs of amyloid or plaques. And we expect these people in blue, who are fast accumulators, to also have faster decline in cognitive abilities than the other groups. The other method that we use to study brain changes during the preclinical phase of Alzheimer's disease is through measuring proteins in the cerebral spinal fluid. This is the data that I work with a lot. Because cerebral spinal fluid is continually being made and clearing out the brain, it gives us a nice snapshot of what's happening in the brain currently. PET imaging is more of an accumulation, a measure of accumulated brain changes, and this is more of a snapshot. One advantage of cerebrospinal fluid is that we can measure a lot of different proteins in a single sample of CSF. And we do. We measure proteins related to inflammation and glial activity, and these are proteins that are produced in response to in response to injury to the brain. We have measures of axonal injury and measures of synaptic dysfunction, so we can try to track neurodegeneration and dysfunction in the communication between neurons in the brain. It's less expensive to get cerebrospinal fluid than to do PET imaging. It takes a little bit less time, but it does require a lumbar puncture. And I know a lot of people are really leery of having a lumbar puncture. So this is the part of the talk where I try to convince you it's really not that bad. I know a lot of people are needle phobic. So the needle that we use to collect cerebrospinal fluid is actually that middle needle. It is incredibly thin. It's an atraumatic needle, which means we can insert it into the tissue without cutting the tissue in any way. We insert the needle between the L4 and L5 vertebrae, which are way down here, which are way below the end of the spinal cord, which is up here. So we're not going to accidentally paralyze anyone when we do this procedure. It's a concern. In our studies, we have people sitting up, and this is my boss, Dr. Cindy Carlson. We always have a doctor or a trained nurse practitioner do all our lumbar punctures, and we always have a study coordinator on hand during the procedure in case the participant needs anything. And then we get a nice clear drip of fluid that looks like this, and we collect about 22 milliliters. Afterwards, we have people lie down for a while, and we get them food. This person looks so happy. This is not actually a participant. This is a picture off of Google, but still. The whole procedure lasts about 40 minutes to an hour. Less than 10% of our participants report side effects. The most common side effects are headache and backache, and these often go away during that rest period right after the procedure. For some people, it persists for a couple of days, but usually any kind of aches or pains clear up with more rest and some over-the-counter medication. If you have been through a colonoscopy, this is a breeze compared to that. The samples are then processed and assayed on a specialized machine. Up to now, we've been sending our samples off to colleagues in Sweden to do all our assaying for us. We now have our very own machine here at UW, so we can start assaying our samples right here on site. We are so excited about that.
I have a question.
Yeah.
About your new machine.
Yeah.
Are you doing some samples in Sweden and here to see if you compare?
Oh yeah, we have lots of planned analyses. We are going to compare samples from Sweden to samples that we do here. We're comparing fresh samples that we assay right away to samples that have been frozen. Yeah, we're looking at day-to-day. I mean, we're doing lots of quality checks. Thank you, it's very important. Yeah?
And the patient is awake for the lumbar puncture?
Yes, so actually the patient is given a little injection of Novocaine at the site. Otherwise, they're totally awake for the whole thing. Yeah.
Can it be done when the patient is asleep, like you would do with a colonoscopy?
No, local anesthetic.
Yeah, we don't ever put the patient out. I don't think anybody would. It's such an easy procedure that I think putting them out would be more of a risk than just doing it. Yeah. Anything else? Okay. So this is an example of data from the Alzheimer's Disease Research Center. The x-axis is disease progression. The brown line on top is the concentration of A-beta-42 in the cerebral spinal fluid sample. So remember, cerebral spinal fluid is cleaning proteins out of the brain, and A-beta-42 is sticky and getting stuck in the brain. So as disease progresses, we actually see a drop in A-beta-42 in the cerebral spinal fluid. And then in contrast, the bottom line, which is supposed to be blue, is a protein called phosphorylated tau 181. And this reflects the formation of neurofibrillary tangles in the brain. These proteins are measured in picograms per milliliter, by the way. They're tiny, tiny amounts. When we get 22 milliliters of cerebrospinal fluid, it's enough cerebrospinal fluid to last us for years. it's a huge amount compared to what we actually need for assaying these things. And again, you can see that signs of amyloid building up in the brain start, this decrease in A beta levels in cerebral spinal fluid starts to happen just a bit before we see a rise in evidence of tau tangles in the brain. So like PET, we can divide people into groups that have high or low levels of amyloid and tau. So these people in the, these colors are so terrible. These people in the top ugly gray box that are high in amyloid and low in tau, these are our healthy seniors. And this sort of pinkish box where people have both amyloid and tau, some of those people are already diagnosed with MCI or dementia. Some of them are not, though. We have some people who are unimpaired in that group. And then we have this group here of people who are amyloid positive only. And this is where having longitudinal data is really key. This is an interesting group of people because some of them will go on to develop tau tangles. Some of them will not. And you can have amyloid in your brain. You can have a lot of amyloid in your brain and never develop Alzheimer's disease or even experience cognitive impairment. So trying to figure out, again, as we're trying to identify people who are on the trajectory to developing Alzheimer's disease in this preclinical phase, it's going to be really key trying to figure out how we can separate people who are going to go on to develop tau tangles from people who are not. This is preliminary analyses. This is hot off the presses. This is using our most recent cerebral spinal fluid data. On the y-axis is, again, that composite measure of cognition measured across multiple time points. So each line is, again, a single individual. And those in the top left quadrant are healthy seniors again. They have no signs of amyloid or tau. Those in the bottom right are those people who have both amyloid and tau. And you can see that, again, we see a lot of bouncing around in cognition. We do see some decline in our healthy seniors. We expect that, but we see a much steeper decline in people who have both signs of amyloid and tau in their cerebral spinal fluid. It's important to note, I think, that cerebral spinal fluid and PET imaging don't measure the exact same proteins, and they don't necessarily tell us the exact same thing, although they overlap a lot. So this is kind of a complicated graph, but on the bottom is a quantitative measure of amyloid plaques in the brain. On the y-axis is a quantitative measure of amyloid accumulation in cerebral spinal fluid. And then this vertical line is the cutoff where we would designate people, anyone above that, as amyloid positive on their PET scan. That's the vertical line. The horizontal line is our cutoff for cerebral spinal fluid, so anyone below that, low is bad. Anyone below that would be considered as amyloid positive on cerebral spinal fluid. And you can see most people are either, not showing an amyloid on both PIB and, scans and cerebral spinal fluid, or they're showing amyloid in both. But we do have this handful of people that look amyloid positive based on their cerebral spinal fluid, but not on their PET scans. That's the point I'm trying to get to. Those people right there. So we're getting more and more evidence that amyloid levels in CSF start to decline before we're actually able to detect amyloid plaques in the brain using PET imaging. So it's really important that we start to use these modalities together. So we spend a lot of time in RAP and ADRC looking at people in this preclinical phase, identifying people in the preclinical phase, and then characterizing them and connecting other measurements that might also indicate who's at risk for progressing and who's not. But the area where identifying people in the preclinical phase is really, really crucial is in clinical trials. There have been many, many clinical trials of drugs for Alzheimer's disease that have failed. And I mentioned there's no cure, and we don't have any treatments that actually get at the root causes, the amyloid plaques and the tau tangles. And one reason that a lot of clinical trials have failed is, people are starting to think now, is because we didn't do a good job of figuring out who was actually at risk for developing Alzheimer's disease and who wasn't. So we're lumping everybody in together and then if somebody's not even at risk, of course the drug's not going to have an effect on them. So as there's advances in measuring these things in cerebral spinal fluid, as there's advances in PET imaging, and as the cost of these things start to come down, we can begin to screen people for those who have actual signs of preclinical Alzheimer's disease before we enroll people in clinical trials. And we're actually doing that right now. We're part of a study called A4. This is a worldwide study of older adults with normal cognition. 5,000 people were screened with PET imaging, and only those who had elevated amyloid levels on those PET images were eligible for enrollment. And this is a clinical trial for a drug that clears amyloid from the brain. The study has finished recruitment, but not all the participants have actually gone through the protocol, and so the results won't be out probably for another year or so, unfortunately. But we're very excited about being a part of that. And we think that this is going to be this system, this way of enrolling people by screening first is going to really help us do a better job of identifying potentially beneficial drugs. So now I want to talk about some of the other research that we're doing here at UW. We do have over 130 faculty, students, postdocs, and staff involved in both RAP and ADRC. It's a huge undertaking, and I tried to count up the number of publications, and then I just gave up. It was too many. And we study Alzheimer's disease from many, many different angles, and I'm using like the global we. So I talked a tiny bit about imaging with biomarkers. We can also use magnetic resonance imaging, a different type of imaging, to look at structural changes in the brain, including size of different brain regions. So we know, for instance, that the hippocampus, which is a region related to memory, is smaller in people with Alzheimer's disease than people without Alzheimer's disease. I touched a bit on using cerebral spinal fluid biomarkers. There's also a huge push to develop ways to measure these proteins in blood. It's much easier to do a blood draw in clinic settings than to get a lumbar puncture. It's also easier to get a blood draw from people from underrepresented groups than to do lumbar punctures with people from traditionally underrepresented populations. I have colleagues who are studying these really early signs of cognitive change, so just as somebody's starting to skew off from what we would consider normal aging, either through formal testing or through looking at speech patterns. I don't have my slide. We also have people who are doing, I'm bringing this up, we also have people who are doing studies on metabolomics and genetics related to Alzheimer's disease, and we have a microbiome study. And I wanted to bring it up because I was just talking to a graduate student this morning who's using the gut microbiome data in his dissertation. And so I was like, oh, I'm glad I ran into you. Tell me what you're finding. And what they're finding is that there's differences in the species of, I don't know what you call it, biota, in the guts of people with Alzheimer's disease compared to people without. And there's also a reduction in the diversity in people with Alzheimer's disease. I mentioned that one of our main goals is to identify factors that can reduce the risk for Alzheimer's disease. And so we spend a lot of time looking at physical activity, lifestyle, vascular risk factors. So for instance, Dr. Ozioma Okonkwo, who's one of our faculty, looks at physical activity. And he found that fitness is actually the ability to sustain a high level of exertion, was related to blood flow to the brain, which is considered to be a measure of brain health. But physical activity, like how much you actually walk around, was not related to blood flow to the brain. He's also part of a multi-site clinical trial called EXERT. This is a trial of the effects of exercise on people who are traditionally sedentary and also have mild cognitive impairment. And the hope is that by getting these people to exercise, we might be able to improve their cognition or at least slow the decline in cognition. Dr. Amy Kind is another one of our faculty members, and she has created a map for the whole United States that measures what we call the area deprivation index at the census level. So here I'm showing just the state of Wisconsin. Areas in blue are areas, high resource areas. Areas in red are low resource areas. The Area Deprivation Index takes into account 17 different factors, and it's things like number of single-headed households, number of people living in poverty, number of libraries, all kinds of things go into this score. And another one of our graduate students recently published a paper showing that the hippocampus, that area that's related to memory, is actually smaller in people from low resource areas compared to people from higher resource areas. And then in the Carlson lab, we're very interested in how vascular health contributes to risk or resilience to cognitive impairment. Actually, this is true of the whole ADRC. There's a lot of us interested in vascular health. So for instance, we have a paper published that shows that people with mild cognitive impairment who also have amyloid in their brains have less blood flow to the brain than people who have MCI but don't have amyloid, so they don't have signs of Alzheimer's disease pathology. And so those people who have lower blood flow to their brain are more likely to progress, we would expect, than people who have better blood flow to the brain. What we're finding, though, is that vascular risk factors have a very significant impact on cognitive impairment, but it seems to be mostly independent of Alzheimer's disease pathology itself. So there's these, like I said, there's multiple roads to dementia. Cindy is also conducting an 18-month clinical trial of the effects of a prescription fish oil through the VA. We are still enrolling participants, so if you know a veteran between the ages of 50 and 75 who's eligible for VA services, who might want to be in a clinical trial, please come see me afterwards. So that was really kind of a whirlwind look at everything, but I do want to kind of talk a little bit about what you can do for yourself. This is a slide that I got from my colleague Kim Mueller, and this is based on a review paper of modifiable risk factors. So the top is showing things that increase your risk of cognitive impairment, not necessarily AD, but cognitive impairment. And on the bottom are things that decrease your risk of cognitive impairment. And the bottom line here is that anything that damages your brain is going to increase your risk for cognitive impairment. Anything that restricts blood flow to the brain, any kind of insults like traumatic brain injury, things like smoking and diabetes, these are things that increase your risk. Things like physical activity, there's some evidence that eating a healthy diet that's high in vegetables and healthy fats actually decreases your risk for cognitive impairment. The bottom line here is that, you know, those things your doctor tells you to do every single time you go in and see him or her, you should be doing those things. Managing blood pressure seems to be really key. Some of you may have heard of the Sprint Mind study. This was a huge trial. This is a randomized clinical trial comparing current recommendations for blood pressure to changing those recommendations to actually a lower level. So we had people who are getting treatment as currently recommended versus people who are actually having their blood pressure lowered to an even lower point. And in that group that had even lower blood pressure than is the current recommendation, there was reduced incidence of MCI and Alzheimer's disease. They didn't actually measure cognitive ability or cognitive impairment in the study because that was not the main outcome measure, but they did have thousands and thousands of people, so we were able to see that decline in the incidence rates. Staying active is always a top recommendation, and there's a program through the National Institute of Aging called Go for Life, which is all about staying active at whatever level you are. If you don't smoke, don't start. If you do smoke, you might want to think about stopping. We have actually a great cessation program here at UW that's helped many, many people, but smoking is a big risk factor for vascular impairment, and vascular impairment is a big risk factor for cognitive impairment. Basically, keeping your body healthy and your mind healthy is going to reduce your risk of vascular dementia and cognitive impairments. And you don't have to wait till this age to start. You can start at this age. or any age in between. So another thing you can do to help is join a research study, and I kind of suspected this wasn't going to work, so I'm going to switch real quickly to YouTube.
My mother gave me a gold coat. Whenever I wear it, I feel close to her. It not only warms my body, but it warms my spirit. She was an educator, a very vibrant, loving Christian woman. When she was diagnosed with Alzheimer's, it was quite a blow to all of us. Alzheimer's doesn't just affect a person, it affects a family. So I really wanted to participate in finding a cure.
It's particularly important for African Americans to engage in research and clinical trials because African Americans are twice more likely to have Alzheimer's disease than any other group.
Put your arm straight out.
Okay. One, there is a fair amount of reluctance in the African American community to participate in clinical trials because of past experiments.
The National Institutes of Health have put into place very strong guidelines that we didn't have, say, 40, 50 years ago. We are engaging all races and ethnicities in our research in a safe way.
I've been participating in clinical trials for 4 years now.
Total, terrible, terrific, tempting.
The benefits of participation are tremendous. You get to know what's going on in the field of science. In other words, what is my hope for not getting a disease like this or for being able to manage it for my loved one?
We found out that they had found a gene that was peculiar to African Americans. I was thrilled to know that I was a part of that. My mother would be very proud of my participation in the research because it's not about me. It's about my family, it's about my community, it's about future generations. The goal is well worth it.
So did anybody recognize some of those backgrounds? That was filmed here. It showed Carrie Gleason walking with her up the stairs, and then one of our nurse practitioners was shown doing some of the strength tests and everything. We were really excited. We got to be part of this advertising campaign. We get excited very easily, by the way, in the Alzheimer's Disease Research Center. There's lots of ways to join a research study. So the Alzheimer's Association has this trial match program. Clinicaltrials.gov allows you to search for clinical trials in your area. Wisconsin, the University of Wisconsin is a site for a program called All of Us. And All of Us is trying to enroll people throughout the United States with the goal of getting to be able, they have multiple goals, but they want to be able to access people's medical records so that we can start to look at things like water quality in an area or air quality in an area and compare that to people's medical records. They also have lots of questionnaires for people to fill out. And like I said, we are still enrolling in ADRC and RAP if you're interested. You can go to our website and look at that. That was a really whirlwind tour, like I said, of Alzheimer's disease and Alzheimer's disease research. So there's a lot of other resources that you can go to. We have a podcast right here in Wisconsin, Dementia Matters podcast. This is with our geriatrician, Nate Chen. It's a fantastic podcast. There's a lot of really interesting research being highlighted in it. But there's also, Nate interviews lots of people and all aspects of Alzheimer's disease. He interviews people who are caregivers, people who are working in memory clinics and in other types of care clinics, and he has a really soothing voice. The Wisconsin Alzheimer's Institute has education as one of its main kind of goals is to provide educational resources, and the ADRC has some educational resources. And both of these groups do a lot of community outreach. It's one of the things that makes me really proud to be part of this organization. We're not just sitting in our silo. We go out in the community. We give talks like this. We have events for our participants. We're hosting an event for the African American community called the Dr. Solomon Fuller Carter Luncheon. And since it's Black History Month, I thought I would talk a little bit about Dr. Solomon Carter. He was born in Liberia, but he got his medical degree here in the United States, and he was selected by Dr. Alzheimer's to go work in his lab. He was a very talented pathologist. He was very interested in dementias, and after working with Dr. Alzheimer's, he came back to the United States. He continued throughout his career to study dementias. And he held a lot of positions that he wasn't technically allowed to hold, but he was able to work through a lot of obstacles to make a big contribution to Alzheimer's disease research. We also have some other events coming up if you want to take a look at our website. And then there's lots of information and lots of resources at the Alzheimer's and Dementia Alliance. Especially for caregivers, this is a really good place to go if you're looking for resources to support you. The Alzheimer's Association has a lot of great educational resources, and so does NIA. And this is a little infographic from Go for Life, all about staying. I don't know, somehow I got on their email list, and they send me emails constantly. And NIA, the National Institute of Aging, really, really loves their infographics. They have so many infographics. I want to thank all of my colleagues, the staff and faculty and students and postdocs who make this research possible. And I also want to thank our funding agencies. We're very fortunate that the state has made funding for Alzheimer's disease research a priority. Nationally, we've been able to get a boost in funding recently. And there's a lot of philanthropic organizations that provide funding for this research as well. And we have amazing participants who come in and let us poke around and ask a million questions and then come back and do it over and over again. And I want to thank everybody for coming. And this is my favorite part of the presentation where I stop talking and you ask me questions. Yes?
Would you expand on the statement you made about exercise and
Blood flow or?
no, what it was endurance versus
Fitness versus physical activity. Yeah, so in this study, people wore an actometer. I forget for how long. Do you know? Yeah, I don't remember. They wore an actometer for a certain amount of time, and then they also did a fitness test on a treadmill. So your fitness is how long you can maintain a certain level of exertion on the treadmill. And that fitness level was associated with measures of blood flow to the brain, but the actual actometer measures were not. And I was kind of scanning that paper, and I'm not entirely certain how to interpret that. Although I do know from other studies of like superagers that fitness is a really big, the fitness level in people who are considered superagers is really off the charts. Yeah?
Is there a particular part of the brain where these pathologies start to appear before it gets to the point of cognitive? Is focusing there? Can you repeat the question?
Yeah, so the question is there a particular area of the brain where these pathologies appear? And for amyloid, amyloid seems to propagate pretty quickly throughout the brain. I think there's some evidence that it starts like in the basal ganglia region and then moves throughout the brain. But oftentimes we just see it appearing everywhere. Tau, on the other hand, definitely starts in the entorhinal cortex and then moves throughout the surrounding regions before moving into the frontal cortex. Yeah.
In the PET scans, where do you get the positrons?
Chicago? I don't know, actually, where we're getting that. I don't. I'm not involved with the kind of nuts and bolts of the PET imaging, so I'm not sure where they get their tracers from. And yes?
You had identified a gene that was a risk factor, APOE?
APOE, yeah.
Okay. How do you identify that in an individual, or do you use that identification in your studies as well?
So we have a couple of, we use a genetic test. So we get plasma from people and we do a genetic test specifically for the APOE gene. And there's a E2, E3, and E4. And E2 is less common and seems to be protective against developing AD. E3 is most common and it's kind of neutral. And then the E4 variation is the risk gene. And we don't use it to, we get it on every person that we have. And we definitely look at whether or not possessing this particular gene modifies any of these other processes. So oftentimes, we'll find that, for instance, vascular risk factors are more pronounced in people with the APOE, the risk APOE4 gene. Yeah.
With that, are you aware of the additional research or any updates on the genes that they've been identifying that protect against the APOE4?
I am not aware of that literature, actually. I only know about the genes that increase risk or definitely cause Alzheimer's disease, like the presenilin 1 and 2 genes. I would love to know more. Maybe you could tell me about it.
I think I many years ago worked in an Alzheimer's research lab, and our neurologist would kind of do lunchtime chats, and she was mentioning that Iceland, because of all the data that they had just recently, probably was about 7 years ago maybe, had identified, maybe even by accident, a gene that seemed to protect these people that had the 4 were not, if they had this other gene, were not getting Alzheimer's disease.
Yeah, I'll have to look into that or ask one of our geneticists, because we do have a few people who specialize in genetics in our group.
Yeah, people are freaking out because they're positive, but we don't even know what's out
Right,
there that
yeah.
could make it so that it doesn't matter.
Okay. Yeah.
So it sounds like from one of your slides early in your presentation that the percentage of seniors who will be getting Alzheimer's or a form of dementia will be increasing as a percentage of the overall senior population, or is the percentage going to stay the same in the forecast?
I think the percentage is expected to stay the same. It's just that we have more people who are living to an older age. However, there's some epidemiological data. It's a bit controversial, showing that because we have so many people taking statins, that we are actually seeing a lower incidence of Alzheimer's disease compared to what we would expect if there weren't so many people taking statins. And this has led to a lot of clinical trials with different types of statins, specifically to see if they improve cognition or slow cognitive decline or have any impact on Alzheimer's disease pathology.
And statins control cholesterol?
Yes.
Right?
Yep.
And what are the results of those studies?
Inconclusive. A lot of statin trials have failed so far, but again, the way people were randomized was not done as well as it could have been. And so there's still people who kind of are holding out hope that if we find people in the preclinical phase, if we do this kind of screening, that we might be able to find that, show that statins are actually having a beneficial impact.
Anything on proton pump inhibitors?
Not that I'm aware of, but I went to a conference specifically for clinical trials, and just about any drug you can think of is being looked at right now in relation to Alzheimer's disease. So probably someone is doing that research, but I'm not aware of it.
There's a study coming out that came out of, I believe, the Federal Republic of Germany, where they took 5,000 healthy 80 90-year-old German males and looked at everything.
Mm-hmm.
You know, the Manhattan graphing.
Right.
But one of the things that was striking that came out was the number of demented men who had been chronically on proton pump inhibitors.
Really?
Yeah.
Oh, okay.
Which,
I'll have to take a
of
look at
course,
that.
we spread around like water here.
Yeah, yeah. And there's a bunch of papers coming out of Mexico City on the effects of pollution on the rates of Alzheimer's disease. So these things could all sort of balance each other out. Like we have higher incidence for this reason and lower incidence for this reason, and we end up back where we started from. Somebody back there? Yeah?
Have anything that you've touched with your studies or touched on to like multi-enzyme therapy?
I'm trying to think back to the clinical trials conference that I went to. I have not seen anything on multi-enzyme therapies. There's been some studies on drug combinations. Not too many, though. It's kind of rare. People really like to look at just one drug at a time. I think it's a lot easier, but maybe from a scientific perspective, not as good. There is something called the finger study or the pointer study, which is actually, it's a behavioral intervention study, not a drug study, but it's a randomized controlled trial where they take people and then try to fix everything they can fix about them. So help them get more active, eat a better diet, manage their chronic conditions to see if we just throw the kitchen sink at this, do we reduce the incidence of dementia? And there was another question back there?
Results? Nothing.
It started in Finland, which is why it's called the finger study, and now it's moving globally to different places around the U.S., but we don't have the results yet. But the initial preliminary results look very good. Yeah. Oh, there's right behind you. Yeah.
You mentioned that you can't make a firm diagnosis until autopsy, but I'm just thinking that if you, in real life, well, and an autopsy, that it's there's often a mixture.
Yes.
Of Alzheimer's and vascular, perhaps.
Yes, absolutely.
And so I'm wondering, how about the PET scan? How definitive is the PET scan? I mean, if you look at it in life, obviously.
I'm sorry.
Well, I mean, we're assuming that people in your study have Alzheimer's disease, but they may not.
They may not. Yeah. So. Oh yeah. So you're talking about people with comorbid conditions, people who have other dementias that might look like Alzheimer's disease in our study, and it's true that we often see when we do a pathology report that people have other brain changes associated with different types of dementias, especially vascular dementia, and there's a couple of reasons that have been proposed for that. One is that the one dementia might make somebody more at risk for the other. Another hypothesis is that we develop these brain changes with aging, and people who get dementia don't stop aging in other ways. They still have other chronic conditions. And one thing that we haven't been very good at is really looking at the whole person, right? Because people, like you said, they don't present with just pure Alzheimer's disease. They have lots of different, especially older people have lots of different problems, lots of things they're taking medication for. And actually, I was just reading this, Dr. Solomon Fuller Carter was very skeptical. Even though he worked in Dr. Alzheimer's lab, he was a little bit skeptical about this thing called Alzheimer's disease being a new disorder because he was seeing in his pathology reports a lot of mixed pathologies. Yeah?
So have you ever found post-mortem that some of your Alzheimer's patients weren't Alzheimer's patients?
I believe we have. I'm not positive about that. But they do a pretty extensive, I actually got to go listen to one of these pathology reports being presented, and they do an extensive look at different brain regions. They look for different types of pathology. They rank them. So it's not even a yes or no, it's a ranking. This is, you know, where we would place somebody in terms of Alzheimer's disease. This is where we would place them in terms of something else. So it's definitely, in reality, it's not quite as clean cut as kind of I've presented here. I think somebody behind you had a question, way back.
I mean, since I've been told you're a statistician, have you looked at Alzheimer mouse models and what is their correlation to human Alzheimer's studies?
Yeah, I have not delved into the mouse models. There are a lot of studies of Alzheimer's disease in mice. We have given mice Alzheimer's disease and cured it in mice many, many different times in many different ways. It's actually not something that occurs naturally in mice, which is kind of interesting. So we have to cause it and then cure it. And I think we've gotten a lot of information, but because it's a very artificial environment, like we are creating pure Alzheimer's disease in mice, and we're not seeing that in people. So I think that the translation from mice to humans has been a bit slow. Yeah?
It sounds like there are two miracle medications, and that is fitness, or exercise towards fitness, and diet. Any major push within the Alzheimer community to increase the level of exercise among seniors, say, or all age groups, and also control diet? And I'm talking about a push involving support and
right, yeah,
and completely holding everything accountable.
Right. So the question was, the statement was that we have physical activity and diet, which seem to be really powerful ways of preventing Alzheimer's disease. And is there a push in the Alzheimer's research community or the Alzheimer's community at large to somehow support better nutrition and more physical activity? The first thing I want to say is physical activity and diet are very, very good for reducing, for improving vascular health and for reducing cognitive impairment. We don't really know how they affect the pathology for Alzheimer's disease. There's nothing that we can point to and say, do this and you will have less plaques in your brain. Do this and you won't develop tau tangles.
You need a longitudinal study.
Right. But yes, we have we're partnering with a couple of different groups, especially within the African American community and the Native American community, to hold classes, exercise classes and nutrition classes. And there's a lot of nutrition and exercise information on the National Institute for Aging. They're supporting a lot of research into exercise and fitness and nutrition. The Alzheimer's Association has a lot of information on these things as well. But in terms of like programs, like Meals on Wheels type programs, I don't know that there's been a lot of policy changes.
For the massive exercise programs in China, for instance.
Right, yeah, we don't have anything like that here. Yeah?
I would strongly suggest going to Dr. Nate Chin's Dementia Matters.
Yes.
I have a great resource through the website,
Mm-hmm.
the Wisconsin Alzheimer's Disease Research Center website, for exactly what you're asking about. He's interviewed the researchers plus practitioners, gerontologists about this.
Yeah, it is a fantastic podcast if you're like going on a car ride or taking the bus or whatever. Pop that on. Dr. Nate Chin.
Nate Chin, thank you.
Yeah, in the back again.
Another question about sensory deprivation of smell, hearing, and
Uh-huh.
sight, and cause and correlation, and how that makes difficulty in your measurements of the patients, aside from the MRIs and
Right.
the other measurements that you're making.
Yeah, we have something that we're working with that was developed by the National Institute of Aging called the Neural Toolkit. And this is a new set of measurements that they're rolling out for all of their Alzheimer's disease research centers. And they include things like your ability to distinguish different smells. It includes a strength test, it includes a hearing test, and a visual acuity test. And they're particularly interested in the hearing test and the smell test, because there's some evidence that as people start to progress in this disease, they actually lose their sensory capabilities. In terms of how that affects like our cognitive testing, certainly changes in vision are going to affect our cognitive testing because a lot of it relies on visual capabilities, but I think that for the most part, if people simply tell us this is hard to see, we make a lot of accommodations to make sure that people can actually do the test that we're giving them.
Any more?
Yeah, one more.
I just was wondering how much you tell the participants in the study. I mean,
Yeah.
if they're CAS, or I mean,
if they're
PET
amyloid
scan.
positive or PET scan, yeah. So the question is, how much do we tell the participant? Right now, our policy is that we do not inform the participant what their scans are showing us. There are other big projects in other Alzheimer's disease research centers that do disclose to their patients. And in fact, Nate Chin, I think, has an entire podcast about this. It's a big question in the research community. And right now, the research on what happens when you disclose to participants what their amyloid state is or other biomarkers shows that people pretty much take it in stride. Most people see it as a positive. Early on, there was a lot of concern that people would get very depressed, maybe even suicidal, if they knew what their amyloid status was, but that doesn't seem to be the case. It seems like people just like to have more information.
Yeah. For some of the studies, you would have to know that you're actually positive in order to get into the study?
Yeah, like the A4 study. Yeah. Because that only enrolls people who are positive. So yeah, in some cases, people know just by virtue of being in the study.
Thank you very much.
All right, thank you, everybody.